welcome to the latest Spotlight On interview. Today I'm joined by Miao and Chien, and we're going to be talking about Miao's work on microscopy-based functional and spatial single-cell sequencing. Miao, if you could please introduce yourself and tell everyone a little bit about what you do.
Thank you, Miao. Yes, so my name is Miao Chien. I am currently an assistant professor at Erasmus University Medical Center in the Department of Molecular Genetics. I'm also a principal investigator at Oncode Institute. So a short introduction about what we are doing. Our group actually developed technologies, specifically single-cell technologies for cancer research. And more specifically, we actually combine multidisciplinary techniques, including advanced imaging, computational analysis, and also single-cell techniques to study the rare and aggressive cancer cells. That's basically what we are doing.
Thank you. So why were you drawn to studying therapy-resistant cancer cells specifically?
Yeah, so that's a good question. So we are indeed interested in those rare and aggressive cancer cells that are responsible for tumor metastasis or therapy resistance. The reason we are interested in that is because we all know at this stage cancers are not really curable. And part of the reason we cannot completely cure those cells we cannot completely cure cancers is after the treatment. There are always a small subpopulations of cancer cells that survive after the treatment, and those small population of cells. Here, I just give them a collective term: aggressive cancer cells. Those cells would just survive after the treatment, and then over a different a period of time, they would relapse and then come back and regrow and even become more aggressively. So that's basically the reason why we are interested in studying those small populations of cells. Yeah.
Thank you. So could you tell us about the challenges in identifying and analyzing these aggressive cancer cells?
Yeah, so people actually are aware, or in the field, people are aware of this type of cells, right? So they know the existence of these cells. But in the past decades, the techniques or the majority of techniques we use to study these cells is to look at specific markers. So if we know the markers of those cells, we can just use, for instance, like FACS sorting, which is a technique you can, use antibody with a fluorescent probe to target those cells, and then those cells can be separated using the machine, which works quite well. And then also people also use this type of techniques to isolate those resistant cells. Not every resistant cell, but quite some resistant cells already. But the thing is, it's not enough, because those cells, not every cells would exist or will express those markers. So just purely using those markers to identify those cells or to study these cells is just simply not enough. And then the way we are studying, or the way the approach we are using in our group is basically to look at those cells not based on their markers, but really look at their behavior. So we really study and analyze individual cells' behaviors, and from those data, we actually can basically, after the analysis, you can narrow down to the subpopulations that are potentially more aggressively, and then we further profile those cells. So the challenging is really to capture those dynamic information, their behavior, basically. So we are talking about the not like static information, it's more like dynamic information, and that is quite often very challenging to do using the current technology. And that to me will be one of the main challenges, and that has been basically tackled by the technology of developing our group. Yeah.
So, like you said, your lab developed a variety of multidisciplinary approaches to investigate these rare and aggressive cancer cells. So Could you give us a quick overview of the different tools that you have developed?
Yes, of course. Yeah, so indeed, as mentioned, so our group is really multidisciplinary, and when I talk about our research pillars, there are normally 4 pillars. So one is advanced imaging. So we actually built our own custom build setup, microscopic setup, to allow us to screen really large quantities of cells. So we are talking about tens of thousands, hundreds of thousands cells in one single field of view. So that is very impressive. And then not only the number of cells, so we can catch this a large amount of cells, the spatial resolution, to look at their individual cells information. We also didn't lose their single cell or subcellular resolution. So this kind of combination is really hard to have using commercial microscopes. And that's part of the reason we built our own setup. So that is the advanced imaging. So the setup, we also implemented a device, no matter what kind of cells you identify, you can also pinpoint to those cells and photo label those cells. And then those cells can then be isolated accordingly. So that is part of the reason we can have this capability to image and then screen a bunch of cells, and then from there identify the subpopulation cells and then isolate them. So that's the first tool we use, advanced imaging. Second part, once we create this, a lot of big imaging data, we also need to have a really robust and advanced image analysis algorithm, because as I also mentioned earlier, we are interested in behaviors of individual cells. So we need to have an algorithm that could help us on the fly analyze individual cells or the data, the image we acquired. During the time-lapse recording. So a really powerful data analysis actually is also very challenging. So for that, we also script our own image analysis to be able to instantly profile the data and instantly export individual cells features. So then we can use that information for further cell isolation. So that's the second part of the tool, and the third part will be single cell technology development. So we use quite a lot of single cell RNA sequencing. Also, we started to use the single cell genomic sequencing and also single cell proteomic profiling as well. And those techniques have to be, those techniques have been developed quite well in the field. And we kind of adapt it and to fit it with our technique, our pipeline. So that's the third part of the tools this is the third tool we use. And the last one will be bioinformatic analysis. So from the data, you can also see we curate quite a lot of data, and the data also quite different than the standard single cell sequencing you will get. So we also need to adapt a little bit of the algorithm, and then to also use that information to dig out the potential targets or, the hidden driving mechanisms that cannot be identified using the current analysis method. So that's basically the 4 different type of tools we use. I realize it's a bit long, but I hope that's clear.
So a lot of multi-omics data integration there,
Definitely.
and a lot of single cell, a lot of spatial, talking about resolution, and also combining that with the image analysis side. And I'm assuming a lot of machine learning and AI on the analysis as well.
Yes.
So could you tell us a little bit about the microscopy-based part of your work, the functional and the spatial single cell sequencing technique?
Yes. So we first developed what we call microscopy-based functional single cell sequencing. So by name, it's basically we can profile or sequence individual single cells based on their functional features. So that's why we call functional single cell sequencing, based on their functional features observed under a microscope. So imagine we basically, so any functional feature you can see, visualize under a microscope, we will be able to isolate those cells. So let's say we screened this 10,000 cells under a single field of view, and then we identify maybe 100 cells that have that display aggressive migration. They display this kind of aggressive migration feature. Then we want to isolate those 100 cells. Then we can just basically use our photo labeling technique I mentioned earlier to label those cells and then isolate those cells for downstream single cell sequencing. The way we, and that is basically the pipeline. So you screen a bunch of cells, and then we do the real-time imaging analysis. You identify the cells you want. And here I use the example of aggressive migration, but any features you can imagine. So we have done, for instance, the cells they displayed or they respond abnormally to the radiation. So then we will look at how their DNA damage response. Their DNA damage response of individual cells after radiation. So from there, you can also see the heterogeneous populations. We can also look at abnormal cell division. So, under the microscope, you can see how cells perform their mitosis. So from those, irregular, regular mitosis features, you can also already see which cells are abnormal, which cells are normal. So then we can also basically isolate those cells that we want to study. So basically any features you can see, or you can see how immune cells escape the cancer cells, or like how immune cells, sorry. You can also see which immune cells interact with the cancer cells, which are not, which can kill the cancer cells, which cannot. That kind of interaction you can also see under a microscope, and then we can also separate those cells selectively. And as mentioned, how we expand, as mentioned, the techniques really to profile cells based on their functional features. So one of the functions we can also think of is the cells, they are located in different spatial location, right? So that's basically how we expand our technology to spatial omics, because under the setup, you can also see where those cells are located, in which area, whether they interact with, whether these cells interact with these cells or not. That kind of information we can also immediately identify. And then basically that can also be used. So our technique can also be used or applied to spatial profiling because of this reason. Yeah.
Okay, thank you so much. So what's really novel about your approach is you're looking at behavioral and functional sort of hallmarks to isolate. So that obviously has an advantage over epitope-dependent forms of isolation, like you mentioned. What about microfluidics? Because that's a big conversation at the moment. Do you take advantage of like new advances in microfluidics in isolating and things like that?
Yeah, so that's a good question. So we currently don't doesn't need a microfluid. So microfluidic is perfect when you don't necessarily need to isolate specific cells with certain markers. So you basically just extract or dissociate your samples into single cell formats, and then you just feed those dissociated cells through this microfluid, and they will be encapsulated in droplets, right? And then in these droplets, they started to to be lysed, and then the RNA will be transcribed. The RNA will be converted to cDNA, and then you create your library for sequencing. So in that setting, that works perfectly if we want to really sequence tens of thousands of cells simultaneously. Right, so then from those tens of thousands individual cells expression profile, you can start to identify different cell types, and then to see what kind of compositions, possible different composition of cells are different in this condition compared to the other condition. So that is basically how people use the microfluidic system. But for us, we don't really need that, because we are more interested in studying the subpopulation of cells that we know they are aggressive, and we want to know what their profiles are. Okay. And using microfluidics, you cannot have that selection power, right? So you just randomly sequence a bunch of cells, but you don't know in the end which cells the cells you profile, whether they express or they display this aggressive features, that you can't link this. So what we do is we first screen under the microscope, and we know which one are the aggressive ones. We selectively isolate those aggressive ones, and we sort them into, we use a play-based sequencing quite often because our cells are quite rare. So 384 well, it's enough for us to study them. So in that case, we basically narrow down to the ones we want, and then we do in-depth sequencing in that sense. So then we don't need to rely on the droplet sequencing. And so that's the difference here.
Okay, thank you. Thank you for elaborating on that a little bit. That's really interesting. So you also use machine learning and AI for your analysis. So could you talk a little bit about the machine learning and AI approach you use, and also maybe what advances in machine learning excite you most, and where you think, whether or not you think it's going to be applied more and more for like data integration and multiomics?
Yeah, it's a very, very good question. So AI is really inevitable. Well, not really inevitable, but it's a really, really useful techniques. But as you also mentioned, it has its own challenging as well. So we do apply AI in our research, and then if you remember earlier, I talked about 4 different pillars in our research group. Currently we implement AI not only in the image analysis part, we also implement that in the bioinformatic analysis part. So for the image analysis, what we do is, when we curate this large image data, we need to really instantly process the data to recognize the cell, detect the cell, and track the cell. And we also need to do that quite accurately, right? Otherwise, we are going to get quite a lot of contamination. So that is part of the reason we implement AI there to improve our detection precision. So this is cell segmentation. So we do implement that part. And another part of AI we actually use 3 parts. So the first one is this image analysis part. The second part is bioinformatics. So many people also use AI or machine learning to basically to dig out the bioinformatic data, sequencing data, and try to see if they can identify some hidden markers or target genes. We also do that, but one of the differences for these, so AI is very smart to identify which shared genes, or which distinct genes across different conditions or across different samples. In our technique, we have the annotation information, right? We know which one is aggressive cell, which are not aggressive cells. So by just by training the algorithm and then by giving this annotation, we actually can more straightforwardly identify the genes that are distinct, that are uniquely expressed in these aggressive cells, other than not in the non-aggressive cancer cells. So this is the type of application we use because we have a simple annotation. There are also quite some group of cells. So some group of people, they when they don't have this annotation, that you can also use AI to try to extract information, but that will be quite challenging and messy because any variation, any differences between sample batch to batch, sample to sample, you started to see a little bit unreliable results basically. So that is how we apply it in our group, but we do see quite some beneficial beneficial outcome results using AI. But the reason it can work for us quite relatively robustly is because we have this very clear annotation at the beginning. So that's also how we applied AI in the bioinformatic analysis. The third one we are recently implementing is to, as I mentioned also earlier, we look at individual cells' behaviors. So quite often we rely on recognizable features like migration, morphology, location. Now we are also training the user AI to train the program, or train the images, or to identify the cells. They haven't displayed this aggressive feature yet. Like they are destined to become aggressive cells, but cannot be identified yet. So we are training this program to be able to identify the cells at early time before they become aggressive cells. And the benefit for this is we can pinpoint down or to get more comprehensive information about how this cell eventually drive to aggressive features. So this kind of thing we are also doing. So this the third type of AI, the application of using AI in our group. Yeah.
So using AI to predict whether a cell is going
Cell
to
fate,
become...
yeah.
Right, that's very interesting. Okay.
Exactly. Yeah, exactly.
So obviously with this AI and your analysis, you're looking at cells that these features are sort of like developing over time. You're looking at different behavioral changes that occur over time. And you mentioned you look at everything in real time.
Yeah.
So do you think more studies will start to use this real-time imaging instead of snapshot? And what advantage does real-time have over snapshot, and what challenges are there in other researchers and other studies adopting real-time imaging analysis?
Yeah, that's a great question. Yeah, so at one of the early questions, I mentioned in the past, decades, people study those aggressive cell or therapy-resistant cells based on markers, and that you can call that snapshot information, right? Static information, which is already very powerful. But what we do is to offer additional information, right? Because some cells, they just don't have these universal markers or known markers. So we started to look at their behavior changes, and that requires real-time imaging. And so the things people can get out of it is to add additional information about those aggressive cells, in addition to what we have studied based on the static information. So I think with this information combined together, it will give us more complete understanding about the driving mechanisms of those aggressive cells. So that is the advantage I think people can get out of it. The challenging part is, it's a real-time imaging. So we're not, not only after acquiring this large image data, one of the challenging elements is for people to apply it is really to have this real-time image analysis algorithm. So we really need to have the real-time image processing algorithm in place. So then they can immediately identify the cells after data acquisition, right? So that instant processing is very important. Otherwise, the target cells you are interested in might already migrate away from their original location, right? So then you cannot separate them. So that is one of the main challenges. But hopefully in the near future, we are actually already submitted a paper, but hopefully in the near future it will be released publicly. We offer basically people or show people how to process these images in a real-time fashion. I hope that will help. And then second part will be, after this real-time image analysis, we also need to be able to, from the data, export the cells you want, right? Based on whatever the feature you define at the beginning, that information has to be further used, for the downstream cell isolation. And you can also start to see more and more. Not very popular yet, but I started to see more and more people started to use this kind of information for cell selection for their own study. But anyway, so in terms of the real-time image analysis, this is these are the challenges I see, and hopefully in the near future, our going to be published paper will help the community. Yeah.
Thank you so much. So just to follow on from the real-time stuff, so you use real-time to analyze the behaviors and this sort of stuff, but there have been sort of advances in temporal analysis for multiomics. So, sort of like live cell recording of transcriptional events that are occurring over time, and not so much with proteomics, but a little bit with genomics and a little bit with epigenomics as well. So do you sort of take measurements at intervals, or do you see like using these technologies, these temporal analysis models, integrating that into your research? Could you just discuss that sort of stuff?
So you're talking about looking at the dynamic transcriptional events? That's what you're saying, right?
Yeah, there's been some advances where they can sort of live record.
Yes. Live record indeed. Yeah, track.
Yeah.
The dynamics of transcription, right? Yes.
Yeah.
So yeah, I actually have one collaborator, in a different institute in the Netherlands. She's actually also looking at that, and we are actually communicating for the collaborative project on this aspect. Yes. So as what also mentioned earlier, like basically anything you could observe under a microscope, we will have a capability to isolate those cells. So in this particular example you mentioned, we are really monitoring the cells that have different dynamics of transcription, right? And that we can also analyze and basically distinguish which population of cells they have a more stochastic transcription properties compared to another group of cells, and maybe it's more stable or less stochastic, something like that. So that kind of information we can also extract from the image analysis, and then ideally we can also isolate those cells accordingly. Yes, interesting question. Yeah.
Thanks very much. So obviously there's also been advances in spatial, increasing resolution and that stuff. There's been a lot of work in that area. Do you think that we've sort of reached a plateau when it comes to spatial, or do you think we could still push that frontier further and there's going to be further advances? What do you think about that?
Yeah, it's definitely not plateau yet. You can see this field, like spatial omics, is really changing very rapidly. Like I would say every half or every year, you can see quite a lot of jump. So I'm actually personally very impressed by the progress in the field. Yeah, so indeed, I think the ultimate goal people want to have for the spatial omics is basically what you can do with the current single cell omics. Right, what are the benefits of the single cell omics? We can profile tons of cells, like tens of thousands of cells. We can have in-depth sequencing profile, let's say tens of thousands of genes per cell. We can have single cell resolution, right? So this is what the single cell current state of our single cell omics techniques can reach. For the spatial omics part, obviously it has a spatial information, but the combination of these three, what I just talked about, a large quantity of cells with in-depth sequencing profile and with single cell resolution, none of the single techniques still have these three combinations yet. These three elements, three-part combination. But that is basically this field is going toward. So if you look at individual one, like for instance, to have a single cell resolution, yes, there are some spatial omics, they have single cell resolution, but they can only profile, hundreds of genes, or maybe a maximum of 1,000 genes, and that's it. Yeah, so it's still different, right? So, but definitely, I have no doubt, like in the near future, people will be able to reach that goal, and including my group, our research, the technique we are developing is basically to combine these three aspects, and without losing their single cell, without losing their spatial information. So that's what we are doing. Yes.
Thank you very much. Okay, so do you going back to your lab's work in your analysis and isolating these aggressive cancer cells, do you still miss some of these rare aggressive cancer cells? And if so, how are you trying to isolate these harder to spot aggressive cancer cells?
Also a great question. So for the phenotypes, we know, and then we observe, and then we isolate. During this whole process, we definitely will lose some cells for sure. So the phenotype we know, as long as after this whole process, screening, isolation, and profiling, as soon as we can have at least tens cells, I would say 50, 100 at least minimum. The more than 100 will be great. If we can reach this kind of number of cells with quality sequencing outcome, we will be able to pull out useful information already. So even though during this process we lose some of the cells during the process, but as long as in the end we can capture at least 50, 100 cells with good qualities or good good quality cells in the end, then we will be able to identify useful information. For the phenotype that we cannot see yet under the microscope, those cells that we will miss, but that is part of things I was talking about when I was commenting on the AI part.
Yeah.
We want to develop algorithm not only based on recognizable features using under a microscope, we also want to have the program that can detect the cells before they display these aggressive features. So then in that case, we can also isolate those rare aggressive cells and then sequence them. So that is something we are also developing and implementing in our lab as well.
Thank you so much. So we talked about sort of where spatial omics is going and how we're trying to get that to the same level as single cell. What do you see as the next frontier for single cell specifically? What's the next step to keep up the momentum?
Yeah, well, so many people already develop great single cell omics techniques as well. I would say it's quite accessible now, but compared to, let's say, box cell sequencing, it's still not that accessible, right? So I think the next frontier in terms of single cell omics only, not spatial omics, I think is to reach the level is accessible as box cell sequencing, as common as box cell sequencing. And not only RNA sequencing part has to be the single cell DNA genomic sequencing and epigenomic sequencing as well, and hopefully with the single cell proteomics as well. So this is kind of, especially single cell proteomics is still relatively new. So to have a perfect combined with this multiple omics at single cell level, I think that will be the next development to make it more accessible for everyone. The second one will be analysis. So there are a lot of well-developed analysis algorithms out there already in the community, but it's still not that how do you say, like, standardized in a way. So that is another part I think people will try to improve as well for the upcoming years. And the last part, I think the development will be truly to integrate that method with the spatial omics. So spatial omics has its own development, and I don't think it reached a plateau yet. But single cell is a little bit in the plateau, but I think the next exciting development will be to bridge these two together. So that will be the three aspects I see for the next frontiers of single cell omics development. Yeah.
Thank you so much. That's all been so interesting. So you're going to be speaking at the Triomic Summit in London.
Yeah.
So why are you looking forward to this Triomic Summit, and what excites you about having a special dedicated event for triomics?
Yeah, so it's if people look at the program, it's super exciting. Like the topics, the people they invite, and it's just amazing. But okay, if I want to give you more specific, the day, not day one, I think it's a focus day on October 18th. There are many talks, not only introducing the advanced techniques, data analysis, but also introduce how these techniques can translate to clinical applications, like how to better understand disease formation, identify hidden targets, hidden therapeutic targets or biomarkers. That kind of talks are basically are covered in that focus day. So and plus many people from pharma companies, from biotech companies are also going to be there to give a presentation. So I thought that it's that first focus day is very very quite exciting for me. The events on 19th and 20th equally exciting because it's not only cover the really most advanced single omics and also spatial omics techniques. They also cover quite a lot of data analysis as well, different methods, and including AI assisted analysis method is also going to be covered in this 2 days. So overall and obviously there are also a lot of network opportunity as well. But overall, I think the program is your program organizer is really put a really exciting program. So I'm super looking forward to it. Yeah.
Thank you. Okay. Thank you so much. My manager is going to be very happy with that.
Yes.
So what what can they expect to learn from your talk at the summit?
Yes. So I think first they definitely will learn this microscopy-based function single-cell sequencing technique, how we can apply it to identify and isolate the rare and aggressive cell populations based on their behaviors or any characteristics. Any characteristics. So we will introduce that technique, and then I will also introduce how we can use that technique to identify otherwise missed targets and molecular mechanisms. And then the last piece will be how we can extend this technique to the spatial omics profiling. So so that I will also talk about how we can combine these methods to really expand to the spatial profiling field, and to really have the capability to profile many cells with in-depth single-cell sequencing and with single-cell resolution. So that is the techniques we are developing, and I will also talk about that technique in my talk as well.
Thank you so much.